Good day. This is the Professopher, CRISPR, PhD. And today is Thursday, December 15th, 2022. It is 12:47 and 47 seconds, a.m. Eastern Standard Time. And today's research, CRISPR technology. So let's hop to it here. The CRISPR technology improves Huntington's disease, symptoms and models. Date December 12, 2022. Source, University of California, San Diego. By directly targeting RNA, researchers were able to eliminate toxic protein buildup that causes the progressive neurodegenerative condition while not significantly disrupting other human genes. The CRISPR is known as a genome editing tool that allows scientists to add, remove, or alter genetic material at specific locations in the genome. It is based on a naturally occurring immune defense system used by bacteria. However, current strategies run the risk of off-target edits at unintended sites that may cause permanent and inheritable chromosomal insertions or genome alterations. And because of this, significant efforts have focused on identifying CRISPR systems that target RNA directly without altering the genome. In the case of Huntington's disease, the condition is caused by repetitive and damaging sequences in the HTT gene. Our cells have a hard time coping repetitive DNA, and these coping errors can cause repetitive sequences to grow longer with each generation. In the Huntington gene, these repeals can sometimes grow to many times their normal length, with the resulting repeal expanded protein tending to aggregate and form toxic lumps in part of the brain called the striatum that is important for regulating movement. The loss of functional neurons in the striatum ultimately leads to Huntington's disease symptoms. The research team investigated whether recently described RNA targeting CRISPR technology could be used to affect RNA, a chemical intermediate between DNA instructions and protein production, accumulation associated with Huntington's disease. They used viral vehicles to deliver the therapy to neuronal cultures, which were developed from stem cells derived from patients with Huntington's disease, and found that the approach not only targeted and destroyed mutant RNA molecules, but also cleared out toxic protein buildup. The research scientists also demonstrated that expression of other human genes was generally not disrupted by the therapy. The goal was to engineer a type of therapy that would only target the toxic RNA that causes Huntington's disease and could keep the rest of the human genome and transcription intact. The research investigators screened the top therapeutic constructs in Huntington's disease patient cell lines to make sure of it. The development of effective therapies for Huntington's disease has proven challenging. In 2021, for example, 2 clinical trials for promising gene therapies were halted following disappointing results performance. As both potential drugs have been touted as game changers for Huntington's disease, currently no treatments can alter the course of the disease, though medications can lessen some symptoms. The Huntington's community was devastated when the clinical trials failed, primarily due to target specificity and toxic effects, but determination has only re-energized the scientific community to find alternative strategies. The researchers found the therapy improved motor coordination, attenuated striatal degradation, and reduced toxic protein levels in a mouse model of Huntington's disease. The improvements lasted for at least 8 months without adverse effects and minimal off-target effects on other RNA molecules. And this is the Professor, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.